Welcome back again, friends. Welcome back to another video session from this channel. So in this video session we will be studying regarding the specific mechanism or the specific process which is involved in organizing the rRNA processing. So what exactly happens during the rRNA processing mechanism or the rRNA processing event? So here, as we all know, in case of eukaryotes, there is an availability of different types of rRNAs. So what are the different types of rRNAs which do exist in case of eukaryotes? The different types of rRNAs which do exist in case of eukaryotes include 5.8S rRNA. Is it clear? 5.8S rRNA, 18S rRNA and 28S rRNA. So all these rRNAs, they are successfully synthesized by taking the advantage of an enzyme, and that specific enzyme, it is nothing but what? RNA polymerase 1. Is it clear? So let me write it on the board. So what it is? RNA polymerase 1. RNA polymerase 1. So here the RNA polymerase 1, it is responsible for the successful expression of 5.8S rRNA, 18S rRNA and 28S rRNA. So here all these rRNAs which do exist in case of eukaryotes, they undergo the processing mechanism or the processing event. But whereas the specific 5S rRNA which is synthesized by taking the advantage of RNA polymerase 3, it does not undergo any processing mechanism or the processing event. Is it clear? So here all these rRNAs which do exist in case of eukaryotes, they are capable to undergo the processing mechanism or the processing event by taking the advantage of certain chemical modifications. Is it clear? So by taking the advantage of certain chemical modifications and as well as by taking the advantage of certain endonucleases and as well as exonucleases. So these rRNAs, they are capable to undergo the processing mechanism or the processing event. Is it clear? So let me write it on the board. What it is? Chemical modifications. Chemical modifications. So here the chemical modifications, they are induced by taking the advantage of an specific RNA which is nothing but what? SNO-RNA. SNO-RNA and as well as by taking the advantage of certain endonucleases and as well as exonucleases. Is it clear? So by taking the advantage of all these events, the specific rRNA, it is capable to undergo the processing mechanism or the processing event. So what exactly happens at the very beginning of the rRNA processing? Is it clear? So for your better understanding, let me present it on the board. So here, let us assume it is the 5.8S rRNA gene. And here, again, it is nothing but what? 28S rRNA gene. And here it is nothing but the 18S rRNA gene. Is it clear? So let me label it. What it is? So here it is nothing but the intron. And again it is nothing but the 5.8S rRNA gene. And here again it is intron. It is nothing but the 18S rRNA, and again it is intron, and here it is nothing but what? 28S rRNA. Is it clear? So here it is nothing but what? The ribosomal double-stranded DNA. So once when the specific ribosomal double-stranded DNA, when it undergoes transcription mechanism or the transcription process, it synthesizes in single-stranded RNA, right? So let me present it here. So here it is nothing but what? A ribosomal double-stranded DNA. So here when it undergoes transcription, is it clear? So once when the specific double-stranded DNA, when it undergoes transcription, it synthesizes in single-stranded RNA. Actually it takes some time to present each and everything on the board. So it is nothing but the single-stranded RNA, right? So here it is simply termed as 45S free rRNA. Is it clear? So here it is nothing but what? The primary transcript which has synthesized during the transcription mechanism. So here the specific primary transcript, it possesses both the exons and as well as the introns. So here the specific introns, they do not have any prominent role, or the specific introns, they do not have any biological activity. Is it clear? So here these specific introns which do not have any biological activity, they has to undergo splicing mechanism, or they has to undergo spliced out. So here in order to organize the specific splicing mechanism or the splicing process, the specific rRNA processing mechanism or the rRNA processing event, it takes the advantage of a specific RNA, and that specific RNA, it is nothing but what? SNO RNA. So what is SNO RNA here? SNO RNA, it stands for what? Small nucleolar RNA. Is it clear? So let me write it here. Small nucleolar RNA. So here the specific SNO RNA, it is responsible for inducing the chemical modifications. Is it clear? So here the specific SNO RNA, it is responsible for inducing certain chemical modifications. So what are the certain chemical modifications which are induced by the SNO RNA here? The various chemical modifications which are induced by taking the advantage of SNO RNA include methylation. Is it clear? Methylation and isomerization. So let me write it here. Methylation, and the other one is isomerization. Is it clear? So here in order to induce the methylation, the specific SNO RNA, it possesses or it contains different types. So what are the different types of SNO RNA here? The different types of SNO RNA include CD box SNO RNA. Is it clear? CD box SNO RNA, it is responsible for inducing the methylation, and whereas HACA box SNO RNA, it is responsible for inducing the isomerization. So what is CD box SNO RNA and what is HACA box SNO RNA? So I will explain you all these things when I explain you about the different types of rRNAs, right? So when I explain you about the different types of RNAs like SNO RNA and snRNA, siRNA, guide RNA. So by that time, surely I will explain you what is CD box SNO RNA and what is HACA box SNO RNA, and how these specific RNAs, they are capable to induce the chemical modifications like methylation and isomerization. But right now we are not much more concerned regarding this concept, because if I explain you the entire concept here, it kills a lot of time. Is it clear? So the reason why, simply I will demonstrate you the specific SNO RNAs how they are capable to regulate the specific chemical modification. Is it clear? So here the specific CD box SNO RNA, it is responsible for inducing the methylation, and whereas HACA box SNO RNA, HACA box SNO RNA, it is responsible for inducing the isomerization. So here what exactly happens here? The specific SNO RNA which is responsible for inducing the chemical modification, it is capable to undergo base pairing with the specific non-coding sequences or the introns. Is it clear? So in order to induce the chemical modification, at the very beginning, the specific SNO RNA, it undergoes base pairing with the non-coding sequences or the introns. So let me print it here. So here it is nothing but what? I. I stands for what? Intron, right? So all these things are nothing but what? Introns. So to the specific introns, the SNO RNAs, they undergo base pairing. So it is nothing but what? SNO RNA, right? SNO RNA. So once when the specific SNO RNA, when it successfully base pairs with the non-coding sequences or the introns, under such condition, the specific SNO RNA, it is capable to induce the methylation. So here, in order to induce the methylation, a specific SNO RNA, which is nothing but CD box SNO RNA, it is required. So by taking the advantage of CD box SNO RNA, here the methylation, it is successfully organized. So what exactly happens during the methylation here? As the name itself suggests, by taking the advantage of methylation, here the methyl group, it successfully recruits to one of the respective nucleotide which is a part of the intron. The specific methyl group, it successfully recruits to any of the specific nucleotide which is a part of the intron. It may be either A, U or G or C. So once when the specific methyl group, when it successfully recruits to the specific nucleotide which is a part of the intron, under such condition, the specific methylation or the specific methylation mechanism, it is successfully organized. And what exactly happens during the isomerization? So here, in order to induce the isomerization mechanism or the isomerization process, a specific SNO RNA, which is nothing but HACA box SNO RNA, it is responsible. So by taking the advantage of HACA box SNO RNA, here the specific isomerization, it is successfully organized. So here the specific isomerization which is successfully organized by the HACA box SNO RNA, it is nothing but what? Pseudo-uridylation. Is it clear? So let me write it here what it is. So let me arrange the things here. Pseudo-uridylation. Isomerization, it is nothing but what? Pseudo-uridylation. So what is pseudo-uridylation? So here if we consider the specific uracil nucleotide, when the specific uracil nucleotide, when it undergoes isomerization, under such condition, the specific pseudo-uridyl nucleotide, it is successfully formed. Is it clear? So let me print it here. Pseudo-uridyl nucleotide, right? So if we consider the specific uracil nucleotide, so here it is nothing but what? The uracil nucleotide, right? And here there is an availability of one hydrogen atom, and here it is the other hydrogen atom, and here there is an availability of ribose sugar. So here it is nothing but what? Uracil nucleotide, right? Is it clear? So once when the specific uracil nucleotide, when it undergoes isomerization, so when it undergoes isomerization, under such condition, pseudo-uridine nucleotide it is successfully formed. So let me print it here. So here it is nothing but what? Pseudo-uridine. Is it clear? So here what is the basic difference in between the uracil nucleotide and as well as pseudo-uridine? In case of the uracil nucleotide, there is an availability of the ribose sugar which is successfully recruited to the nitrogen atom which is present at the first position. Is it clear? So here the specific ribose sugar, it is in association with the nitrogen atom which is present at the first position in case of the uracil nucleotide. But whereas once when the specific uracil nucleotide, when it undergoes isomerization, under such condition, the specific ribose sugar, instead of recruiting to the specific nitrogen atom which is present at the first position, the specific ribose sugar, it successfully recruits to the nitrogen atom which is present at the fifth position. So once when the specific ribose sugar, when it successfully recruits to the nitrogen atom which is present at the fifth position, under such condition, the pseudo-uridine nucleotide, it is successfully formed. So here by taking the advantage of HACA box SNO RNA, here multiple pseudo-uridine nucleotides, they are successfully recruited to the specific introns. So once when the chemical modifications, when they are successfully organized by taking the advantage of different types of SNO RNAs like CD box SNO RNA and HEACA box SNO RNA, under such condition, now here in order to cleave the specific introns, is it clear? In order to cleave the specific introns, certain endonucleases they come into the picture. Once when the specific introns, when they undergo chemical modification by taking the advantage of SNO RNA, under such condition, now the specific introns, they act as a hotspot for certain endonucleases. Now the specific endonucleases, they are capable to recognize, they are capable to cleave the specific introns. So let me write it here. In the presence of endonucleases, in the presence of endonucleases, the specific introns which had undergone certain chemical modifications by taking the advantage of SNO RNA, they undergo successful splicing. So let me erase the thing here. So here it is nothing but the 5.8s rRNA. And here it is nothing but the 18s rRNA, and here it is nothing but the 28s rRNA. Is it clear? So here once when the specific non-coding sequences or the introns, when they undergo successful splicing by taking the advantage of endonucleases, at the very end, now here one more other enzyme it comes into the picture, and here it is nothing but what? The specific exonuclease. So here the specific exonuclease enzyme, it is responsible for inducing the trimming. Is it clear? So let me write it here. Exonucleases, they are responsible for inducing the trimming, right? So here what is the role of exonucleases? So these exonucleases, they play a prominent role in order to induce the trimming at the very ends. Is it clear? That means when the specific endonuclease enzyme, when it successfully cleaves the introns, under such condition, some part of the intron, it may remain in association with the exon. So here in order to overcome the problem, the specific mechanism or the process, it takes the advantage of a specific exonuclease. And here the specific exonuclease, it is responsible for trimming the specific ends. That means the specific non-coding sequence or the specific part of the non-coding sequence which is in association with the exon, it successfully cleaves by taking the advantage of these exonucleases. Is it clear? So here the specific exonucleases, they are responsible for trimming the specific ends. So once when the specific endonuclease and exonuclease, when they are capable to cleave the non-coding sequences or the introns, and once when the specific exonuclease enzyme, when it is capable to trim the extreme ends, under such condition, the specific rRNA processing mechanism or the rRNA processing event, it has successfully completed. Is it clear? So here this is regarding the specific mechanism or the specific process which is involved in organizing the rRNA processing event. So I hope that this video will help you a lot. So if you like this video, just hit the like button and share it to your friends. And I remember you people to subscribe my channel for getting more and more videos what I make day to day. Thank you.